Hello and welcome to our channel. I'm Mrs. Taylor West, and today we're diving into the fascinating world of embryology. We'll explore the early stages of development, including cleavage and blastulation. Whether you're an expectant mother or a biology student, understanding these processes can be both exciting and informative. Embryology is the study of how embryos develop from fertilization to birth. It all begins with fertilization, where a sperm cell fuses with an egg cell to form a zygote. This single-celled zygote contains all the genetic information necessary to develop into a complete organism. Source: Gilbert. Scott F. Developmental Biology, 10th edition. The first major event in embryogenesis is cleavage. Cleavage is a series of rapid mitotic divisions that the zygote undergoes, resulting in an increase in cell number without an increase in overall size. These divisions create smaller cells called blastomeres. The purpose of cleavage is to quickly produce a multicellular structure from the single-celled zygote. Source: Moore, Keith L., and T.V.N. Persaud. The Developing Human Clinically Oriented Embryology, 10th edition. As cleavage continues, the cells form a solid ball called a morula. Around day 4 after fertilization, the morula transforms into a blastocyst through a process known as blastulation. During blastulation, the cells start to differentiate and form a hollow sphere with a fluid-filled cavity called the blastocoel. The blastocyst consists of two main parts, the inner cell mass and the trophoblast. The inner cell mass, located at one pole of the blastocyst, will eventually develop into the embryo itself. These cells are pluripotent, meaning they have the potential to differentiate into any cell type in the body. On the other hand, the outer layer of cells, called the trophoblast, will contribute to the formation of the placenta and other supporting tissues needed for fetal development. The transition from morula to blastocyst is critical because it marks the beginning of the embryo's ability to implant into the uterine wall. Implantation occurs around day 6 to 7 after fertilization, when the blastocyst attaches to the endometrium of the uterus. The trophoblast cells play a vital role in this process by secreting enzymes that allow the blastocyst to penetrate the uterine lining and establish a connection with the maternal blood supply. During implantation, the inner cell mass undergoes further differentiation to form the bilaminar disc, consisting of the epiblast and hypoblast layers. This stage sets the foundation for the next phase of development called gastrulation, where the three primary germ layers, ectoderm, mesoderm, and endoderm, are formed. These layers will give rise to all the tissues and organs of the body. Understanding these early stages of development is crucial for expectant mothers to appreciate the complexity and wonder of human development. For biology students, these processes provide fundamental knowledge about how life begins and progresses at the cellular and molecular levels. Thank you for joining me today. If you found this video helpful, please subscribe to our channel for more educational content. Be sure to check the description below for references and additional resources. See you next time.